I'm Joanna Long. I'm a professor of biochemistry at the University of Florida, and I'm also an associate director at the United States National High Magnetic Field Laboratory. Well, so we are still working on the pulmonary surfactant. I am pretty sure I'll be working on it my entire career because now what we finally figured out was the role pH plays and what the lipids are doing, and now we've gone back to looking at these fairly simple proteins. They're not very large, but they have kind of this very specific way of interacting with the lipids to make them very sensitive to what position they are in the lung and what lipids they're interacting with so that they can have this function where they're actually sorting the lipids. And we're really interested in this, how do you sort lipids? Because when you look at biological membranes, they actually are highly specific, and that's based on their lipid composition. And when we first started thinking about this problem, we were thinking about cells, but cells are highly complex with literally thousands of different types of lipids. Whereas pulmonary surfactant is very simple with 4 basic lipids in it. I think the most powerful thing about NMR is first the fact that you can, if you isotopically enrich or you use certain quantum mechanical selection strategies, you can pinpoint a molecule in its native environment. And you can do it non-invasively. I actually have had my children go in my MRI scanner several times because they like to volunteer for research. And I never worry about the safety because it's such a non-invasive technique. That's the real power of it. But then when you think about the fact that it's non-invasive and yet at the same time you understand things at atomic resolution and you understand the chemistry, not just, you don't just get a pretty image, but you get to understand the chemistry. That's also very unique about magnetic resonance. I study proteins that traffic lipids, and we got interested in looking at pulmonary surfactant, which is a highly specialized fluid that allows us to breathe at ambient pressures. And, when we first started, we were thinking, we just want to see what this interesting protein does. And then as we started studying the system, we realized actually the lipids themselves are doing some really interesting choreographic movement as they come out of the cells and go up to the air-water interface. And there's this whole timeline of how things happen. And some of it is, what lipids are produced and in what quantities they're produced, but then the proteins play a certain role. And then lastly, we realized only recently the role that something as simple as pH plays in the whole choreography of what's going on. And it's because, we're breathing in oxygen and then we, breathe out carbon dioxide, and the carbon dioxide actually lowers the pH in the lung. And so nature has this ingenious way of actually using the fact that the pH is changing in the lung to then give pulmonary surfactant its special function. In DNP, there are actually, I have the pulmonary surfactant system where we're now able to see these subtle differences in protein secondary structure that kind of explain how it's able to be a sensor in terms of lipid environment. And then I have a second project where I'm working with a collaborator that studies enzymes that synthesize amino acids. And these are enzymes that they have known the structures of for many years because of X-ray crystallography. But at the end of the day, how those enzymes work is they're moving hydrogen atoms from one molecule to another. They're literally doing chemistry in the active site. And that wasn't fully understood even though they had full X-ray crystal structures, because X-ray crystallography, you see larger atoms like carbons and nitrogens and that sort of thing, but it's very hard to see where the hydrogens are, and they're the ones doing all of the important work. So DNP has enabled that research. So in the specific systems I study, it's more kind of a basic understanding, but in some of the collaborative projects I'm involved with the National Magnet Lab, it's bringing new technologies into the clinic for diagnosing and staging cancer. For my university is one of the leading universities in terms of using magnetic resonance technologies to evaluate gene therapies. So we studied muscular dystrophy, and we're able to actually quantitatively look at the effects of gene therapy and evaluate whether the strategies they're following are actually effective before you ever see the long-term symptoms of like full disease manifestation. So, and that's all done with magnetic resonance, and it's done non-invasively. And it's, this is research on young children that are coming into the magnets, and they're able to stage and watch the progression. And that's because what we've developed using NMR in terms of looking at chemistry is now to this stage where you can actually do some of those experiments in living humans.